introduce our first keynote speaker of today, who's Jessica Lu, who I personally know. So she works for NOAA at the Geophysical Fluid Dynamics Lab, so GFDL, and she's been working on developing the next generation of marine ecosystem models. Her other interests are gelatinous zooplankton, submesoscale plankton dynamics, and today she's going to present a very exciting talk. Jessica, may go ahead.
Thank you very much. I'm very happy to be here today. And let me just get my slides together. All right, thank you very much for the invitation. All right, I'm happy to present some work on modeling the global scale ecological and biogeochemical impacts of gelatinous zooplankton. So as Gabby introduced, I am at the NOAA Geophysical Fluid Dynamics Laboratory in Princeton, New Jersey. I put my email down here. And this work was a collaborative effort with a number of people, including Charlie Stock and John Dunn at GFDL, a number of individuals from the Global Jelly Working Group, Rob Condon, Kathy Lucas, who is actually at NOC, Kylie Pitt and Carlos Duarte, and then also Natasha Hensky at UNSW and Bob Cowan, Oregon State. So the marine biological pump, as you all probably know, is a collection of biologically mediated processes that serve to transport organic matter from the ocean surface to depth. Models and observations typically estimate that its strength ranges about an order of magnitude from 5 to 12 petagrams of carbon per year. And it's also projected to decline between 1 to 12% by the year 2100 under a high emissions scenario. These are just from CMIP-5 models. Of the processes that comprise the biological pump, the likely largest sources of particulate organic carbon come from phytoplankton aggregation, the sinking fecal pellets of mesozooplankton, and the respiration and excretion from diel vertical migration, and then some unknown sources from gelatinous zooplankton. But what is notable is that these all may have different trajectories under climate change. So Earth system models typically only represent a fraction of these processes, the phytoplankton aggregation and the zooplankton egestion amongst them. And just as a note, this figure is from Steinberg and Landry 2017, and it was the first time in a major publication that did the biological pump schematic include these possible gelatinous zooplankton. And at the time, the quantities and contributions from the GZ were really quite unknown. But because of their ecology and physiology, they were hypothesized to be potentially very globally significant. So in this talk, I'll start with a short overview of the gelatinous zooplankton and some factors that have contributed to them being understudied compared to other zooplankton. And next, I'll give a short presentation of the data-driven study assessing the impact of 3 different types of gelatinous zooplankton in the carbon cycle. And then I hope to focus the bulk of my talk on some new work incorporating gelatinous zooplankton into ocean biogeochemical model Cobalt, the NOAA GFDL ocean biogeochemical model. And at the end of this talk, I hope to convince you that gelatinous zooplankton are globally significant contributors to biogeochemical cycles and should be incorporated in future Earth system models. So gelatinous zooplankton, as shown here in this figure on the this image on the left, are an extremely diverse class. These include the jellyfish, kind of the cnidarians and hydrozoans, the comb jellies, and then also salps, which are these pelagic tunicates. But they're just found throughout the world's oceans and are characterized by these fragile watery bodies with a low carbon content. The gelatinous body plan provides some very distinct advantages where they can achieve these large sizes without the corresponding carbon content. This allows them to have more effective passive feeding, escape predation, and then also many species produce asexually. So they have these very large blooms. But unfortunately, there has been a number of barriers to their study. They've been historically very difficult to sample using traditional methods such as plankton nets, and also thought to be trophic dead ends. So in ecosystems where the studies have mostly been focused on energy flows to fish, they've been relatively ignored because they have been thought to be unimportant. And then also, as I mentioned, they have unknown importance to the ocean biogeochemical cycles. So jumping into a few of these barriers, the first are plankton nets, which result in systematically biased sampling for gelatinous zooplankton. So plankton nets are shown here and have been used for over 100 years as the predominant way to sample plankton in the ocean. But in this one paper from Remsen et al. 2004, in a comparison, side-by-side comparison between a plankton net and a towed imaging system, they showed that for crustacean zooplankton, on these, such as the copepods and krill, euphausids, there is virtually no difference between the carbon biomass caught by plankton net and by the imager. But for gelatinous zooplankton or soft-bodied zooplankton, there is a huge difference, up to maybe an order of magnitude or so. And this poses a big problem for us because we've based all of our validation data, all of our basically our data kind of estimating the role of zooplankton in marine food webs on predominantly plankton net-based sampling. And then the second is that the gelatinous zooplankton have historically not been thought as important as prey for higher trophic levels. So in terms of gut digestion time, the gelatinous zooplankton are digested very quickly in guts compared to their crustacean counterparts. But newer technologies such as DNA barcoding, also animal-borne cameras, and plankton imaging systems, for example, have shown that there's a wide range of organisms that predate on gelatinous zooplankton, and they really aren't the trophic dead ends that we previously have thought them to be. So the first study that I wanted to tell you a little bit about was one that was published last year in Global Biogeochemical Cycles, titled Gelatinous Zooplankton Mediated Carbon Flows in the Global Ocean, a data-driven modeling study. And this is an effort where we constructed an offline model based on observations of data to estimate, are these gelatinous zooplankton potentially important? Are they worthy of additional study in terms of global carbon cycles? So in this data-driven study, what we did is we constructed a model of that represented the top 200 meters of the water column and export to depth in these 1D columns. So in the top 200 meters, what we started with the GZ kind of biomass and then estimated based on their biomass and a set of allometric equations, how much prey are they consuming, and then how much is going to respiration, how much is going to egestion, how much is lost for reproduction, and then how much goes to predators, and then lastly, how much gets contributed to particular organic matter. And then with this particular organic matter that's produced, then we estimate how much sinks to various different depths, most notably past 100 meters, 1,000 meters into the seafloor. This is just the data that we started off with, showing that just in terms of biomass, the gelatinous zooplankton comprise about 0.5 petagrams of carbon as a standing stock. We've kind of divided their biomass into kind of different regions, from coastal regions, low chlorophyll, so these are the gyres, to the seasonal seas, high chlorophyll seasonal seas, and the permanently stratified regions, and just shown just the degree of variation in these different biomes, as you can also see here. In terms of their sinking, we've used their sinking rates from a number of studies, including this one from Labrato et al. 2013, showing that gelatinous zooplankton sink very quickly through the water column compared to other typical particulate organic matter that might sink between 50 to 100 meters per day. They can actually sink up to from 500 to 1,500 meters per day, such that a bloom of jellyfish will die and sink to the bottom of the ocean with completely intact. So within a couple days, they're not degraded at all, and they just kind of sink like rocks through the water column. So we wanted to see what effect these might be. Oh, sorry. Before I move on from this slide, I just want to say that I'm not going to present the full details of this study. We did a lot of uncertainty quantification here, and I recommend that if you have more questions, to either email me or check out the publication. So we did a number of single point validation for this model and showed that in general, we had a couple different cases of low biomass and medium biomass and high biomass, that the observations generally, except for a few circumstances, matched this offline model. And then so here are the results for the global fluxes. These are a lot of numbers, so I actually would like you to kind of focus on this top left box, where all the units here are in petagrams of carbon per year. So in general, we found that the gelatinous zooplankton will, as a whole, consume a lot of carbon. Just in this offline model, we estimated that they consume nearly 11 petagrams of carbon per year, of which a small fraction of it gets respired and sent to dissolved pools, but a big majority of it gets produced as particulate organic matter and then kind of sinks. But what is notable here, if you just ignore kind of the massive numbers here, what is notable is that these salps, these pelagic tunicates, contributed about 77% of the carbon export, even though they were a relatively minor fraction of the gelatinous zooplankton biomass. So this kind of indicated to us that of the three major types of gelatinous zooplankton, the jelly, the true jellyfish, the comb jellies, and these pelagic tunicate salps, that it might be most important to focus on these in terms of if we wanted to understand their contribution to global biogeochemical cycles. However, there is a huge caveat with this study, is that the offline models do not take into account food web and biogeochemical feedbacks. So, we did a lot of checks in this offline study to make sure that the numbers were not completely out of bounds. But again, if you're consuming so much prey, then it will have downstream impacts. So we wanted to then take this to the next step and then incorporate these gelatinous zooplankton into an online model. So that is what we did. So this is work that I'm working on right now, and I would appreciate feedback from this group. We're planning to submit it to Progress in Oceanography, hopefully early next year. So this is online modeling of gelatinous zooplankton with cobalt, and cobalt is the ocean biogeochemical model of GFDL. And as you can see here, it represents plankton ecosystem in 7 major groups: bacteria, heterotrophic bacteria, free-living bacteria, small phytoplankton, diazotrophs, large phytoplankton, and then 3 different sizes of zooplankton, including these medium and large zooplankton that are primarily modeled as crustaceans. So as I mentioned in my previous slide, we were interested in these pelagic tunicates because of their potential unique niche and disproportionate role in biogeochemical cycles. We thought that, so the pelagic tunicates, kind of digging into them a little bit more, are also a diverse group that includes, these salps, also their smaller counterparts, the doliolids, and these much larger colonial pyrosomes, as well as these the appendicularians, which I like to call kind of the spiders of the ocean. They kind of create these 2 sets of like mucus mesh nets to capture their prey. These organisms will consume prey at a very high predator-prey length ratio, as you can see here. These are the pelagic tunicates as compared to copepods or ciliates or flagellates. They're consuming at a higher clearance rate at a much, I guess here is like the prey to predator length. So their size compared to the size of their prey are gargantuan. They are kind of the gargantuan feeders in the ocean. So we thought that they potentially might have a role in mediating export from the microbial loop, actually. So in this work, we had 4 scientific questions. The first is, so if we're accounting for systematic undersampling by nets, if nets are undersampling gelatinous zooplankton by order of magnitude, can we still capture these large observed gradients in pelagic tunicate biomass in an online model? And then second, can we reconcile this expanded role of gelatinous zooplankton with established evidence for the importance of crustacean zooplankton? So there's been decades of study showing the importance of copepods, krill, and other crustaceans in marine ecosystems. Can this expanded role of GZ be reconciled with that? And then thirdly, very simply, how does the online estimated GZ mediated export compare with the offline measurements? And lastly, what is their impact on the partitioning of carbon flows to recycling, export, and flux to higher trophic levels? So in this study, we modified the cobalt food web by adding in like a small tunicate and a large tunicate group. What is important to note is that they are able to feed on a wide range of organisms, but actually may not feed very well. So they are, they're often not super selective compared to other zooplankton in the food that they predate on. So I've implemented this as lower prey selectivity. And then they are also they also are predated upon by a number of other organisms, such as crustacean, mesozooplankton, and the higher trophic levels predators. We modeled these gelatinous zooplankton considering a number of key trait trade-offs. I'm not sure if your class kind of talked about the trade-offs that we use to model, phytoplankton and zooplankton, but these end up being really important to represent distinctive ecological niches of new classes that we might introduce into the model. So as I mentioned, the pelagic tunicates, the pelagic tunicates are able to feed on multiple trophic levels, including picoplankton, compared to the crustaceans that are more targeted. They have a wide range in predator to prey size ratios and higher clearance rates. And they are also notable for having higher just basal respiration rates. And this just means at rest, they just have a higher respiration rate because they are continuously filtering. They're filter feeders. However, there's a few downsides, is that they're, because of their continuous filtering, their filter can clog, and the appendicular house can also clog up. And so this decreases their assimilation efficiency at high food concentrations. There are also this aggregation mortality and mass die-offs, kind of representing the kind of massive falls of jelly falls from the surface to depth. And then they are important as prey for higher trophic levels, but comparatively to other crustaceans, they're maybe a little bit less attractive. So here on the right, I've plotted their kind of like an idealized specific production at 25 degrees C, showing that compared to, kind of the microzooplankton, the medium and large crustacean zooplankton, the pelagic tunicates are kind of falling in between the three existing zooplankton types. All right, so and then we compiled data from a number of different sources, as listed here, and plotted them on these maps. And then notably, what we noticed is when you plot the log of their biomass compared to surface chlorophyll, there is a very distinctive pattern that contrasts between tunicates versus mesozooplankton. The first is that you'll see that there's a lot of variability in the data, and that might just be due to lower sampling overall. But particularly in the large tunicates, as chlorophyll increases, the scaling slope between the biomass and chlorophyll is actually relatively flat. This implies that, as climate change is projected to, increase stratification and decrease production, decrease the size of the mean plankton, that their role in marine ecosystems may actually increase relative to mesozooplankton. So we wanted to be able to capture this contrast. This kind of represents like a large scale contrast between biomes, and ended up being one of the key validation metrics for our study. So after a bit of development, we ended up with a simulation. So we ran this just in a core forest. Using, sorry, I'm going to move this so I can see. So we used the MOM6 SIS2 physical framework with GZ Cobalt. We ran this for 60 years using core 2 forcing. And the results here are showing the climatology of the last 20 years of the simulation. So what you can see is that there is relatively pretty good agreement between the observations and the model, where you can see higher biomass kind of in these regions, and compared to slightly lower biomass, kind of in the gyres and in the less productive areas. But actually of note is that there is less contrast in the large tunicates compared to the small tunicates. And then of our primary validation metric, the model was able to capture this emergent relationship that I described, where in the large tunicates, as you can see, the black dots are the model compared to the blue dots with the observations. We were able to, to match the observations quite well. This was not the case in the small tunicates, and I don't really have time to discuss this too much, but overall their biomass was about an order of magnitude less than the large tunicates. So this ended up being a slightly lower target for us than capturing this large tunicate versus the large mesozooplankton contrast. Very, very quickly, these slides are just to show that the difference between the new GZ cobalt simulation compared to the control our differences are almost negligible, and also match the observations very well in terms of surface chlorophyll, nitrate, phosphate, and silicate. And all of the observations are from the macronutrients are from the World Ocean Atlas, and chlorophyll is from CWIFS and MODIS. In terms of the seasonal cycle, what I've done here is then again broken up into these large regions of the ocean. You can see that in general, so what I have here, I'll just orient you really quickly. I have small zooplankton, medium, and large zooplankton. These were the original 3 classes, and these are the 2 new classes that we added. And I adjusted the seasonality so that all summer months are matched between northern and southern hemisphere. So I shifted the southern hemisphere about 6 months. And what you can see is that compared to the cobalt control in this dashed line and the GZ cobalt is and the solid line, the introduction of the 2 new gelatinous classes reduces the zooplankton biomass. I mean, this kind of makes sense. They are competing with the other zooplankton. So the other zooplankton biomass kind of reduces accordingly. But the biggest differences are in these purple regions, the permanently stratified, kind of these upwelling regions, particularly in the small zooplankton and this microzooplankton. Of note is the large tunicates have a different seasonal cycle, in the contrast between the permanently stratified and then the seasonally stratified, where their highest biomass was not actually in the seasonal seas. Their highest biomass was in kind of these upwelling areas. And this is also in contrast with the small tunicates, where their biomass was about order of magnitude less, and their highest biomass was in the seasonal seas. So this is now, so I think that this is potentially the most important set of plots here. So now what I've done is then contrasted the set of processes that go into kind of recycling versus export. So here, just to orient you again, on the first row, I've plotted microzooplankton production over NPP. And the first column is the GZ cobalt. The second column is the cobalt control. And then the third column is the difference between the 2. The second row, I've plotted all mesozooplankton production divided by NPP, and the fate of this these mesozooplankton go to export and higher trophic levels. Third row here is export flux over NPP, or the PE ratio, and this obviously indicates export. And then the last row here is bacteria production over NPP, and the fate of which goes to recycling. So what you will notice is a very clear contrast between processes that go into recycling, so the bacteria production and the microzooplankton production, compared with the processes that go to export. So the introduction of these pelagic tunicates, these gelatinous zooplankton, shifted the balance of the ocean towards more export compared to recycling. And notably, they're competing quite strongly against the microzooplankton. So just in terms of like a total bulk amount, this is export production. The total export production was increased about 1.2 petagrams, but this translated to only, about 0.4 petagrams of increase in actual export flux out of 100 meters. We didn't consider just in this study the differences in the sinking rate, just because in the top 100 meters, those differences result in pretty low differences in flux. But what you can see here is that the introduction of the pelagic tunicates, or the gelatinous zooplankton, resulted in kind of a redistribution of the export production away from the small mesozooplankton and also to a smaller degree the large mesozooplankton towards these tunicates. So and then when we add these numbers together compared to the offline model, this is about 2.7 petagrams of carbon in terms of export flux, compared to the offline model, which estimated about 3 to 4.5 petagrams of carbon in the top 200 meters. This is within range, but slightly on the lower side. But the key point here is that when we add in these new groups, that it really just repartitions the export production, but they are quite significant globally. And then this is my last slide before the conclusion. In terms of their contributions to higher trophic levels, so this is like flux to fisheries and et cetera, compared to the cobalt control, this was fairly comparable, the new GZ cobalt. And again, their estimates were slightly on the lower side compared to the offline model. And the introduction of the GZ resulted in kind of a redistribution of the fluxes to the higher trophic levels, particularly from the small mesozooplankton. All right. Hopefully, Gabby, I haven't gone over on time. I haven't heard a time check. But just in terms of my conclusions, so many empirical studies hint at this expanded role for GZ in marine food webs and biogeochemical cycles. And we were able to create a simulation that captures these large-scale emergent patterns while accounting for this systematic undersampling. It satisfies a range of biogeochemical constraints and maintains this robust crustacean zooplankton population. Results show that the gelatinous zooplankton are significant competitors with microzooplankton, particularly in upwelling areas. And the net biogeochemical impact is to shift the system away from recycling and towards export. Their contributions to export are in the middle, roughly middle of the offline estimates, but are on lower end in terms of the fluxes to higher trophic levels, and results in repartitioning of export from other sources. So I will leave this conclusion slide up, and I'm happy to take questions if my time isn't already spent. Thank you.